So this is a video about the history of pioneers of protein structures who basically developed methods using X-rays mainly to work out structure of protein. So it's protein structure pioneers. Um, so started with Bragg in 1912, who with William and Lawrence Bragg, they both got Nobel Prizes, Copley Medal. They were, I think one of them might order a medal. They were very eminent, very famous scientists. Invented X-ray diffraction and crystallography, but they discovered X-ray diffraction. You didn't invent it, but they discovered it. And they discovered the crystallography as a result of that. Astorbury worked out the triple helix structure of keratin. Hair in 1931, and then Burnell, I think it was John Burnell, 1930s, worked on the first peptide structure. Peptides are amino acid chains running from 2 to a few hundred. And then Dorothy Hodgkin, who's one of the most famous scientists in the 20th century, started insulin in 1930. I believe it took over 30 years to finish it. She did that to 2.8 Armstrong resolution. Linus Pauling, the American chemist, worked out the structure of alpha helices and beta sheets in 1951. John Kenru worked in myoglobin in 1961, worked out its 3D structure. It was the first use of computers in crystallography. He got that to 2 Armstrongs. This myoglobin has over 1,000 amino acids. Max Prout's one of the heavyweights. So he worked out hemoglobin structure, just 5.5 Armstrongs. the smaller the number, the more detailed these basically the images that you get, the model. Atoms of size of hydrogen's about 1 angstrom diameter. So he started that, and although he got his PhD after 3 years, it took him 16 years to finish the work. He had to invent techniques like isomorphic replacement, where you replace atoms with their isomers, heavy isomers, and it started in 37, finished it in 1953. He got Nobel Prize, Copley Medal, Royal Medal, all in merit. Some of these people are incredibly eminent. But then they changed the face of the subject, completely rewrote all the textbooks, and made pioneering work to biochemistry. David Phillips works in lysozyme, the first enzyme. '3D structure. That was in the '60s, I think. Ramachandran plot in 1963. So he worked out a plot of phi versus psi angles. That means in amino acids, the phi angle is the primary carbon to carbon bond, and that's the primary carbon to nitrogen bond in the amino acid structure, okay? And that shows you where the energy is, how it's distributed. Fred Sanger. Which shows the angle, how angles and energy are distributed. Fred Sanger invented protein sequences in 1951-52. Amundsen, 1961, came up with a hypothesis sequence to 3D structure. More modern times, this is so you plot the angles, so it shows you the angles and the energetics of them. Borthreich NMR used NMR on small proteins in the 70s and 90s, which is pioneering. And then Henderson recently got the Copley Medal, and Frank and Dubujet. invented I think it means cryogenic, cryo-electron microscopy, which basically involves no crystals. So when you have very big proteins or asymmetrical ones, it's very difficult often to create proteins. Sorry, to create the crystals is very difficult to achieve. Very difficult to create crystals, and so this avoids that. Venki and Ada Yonath. In recent times, they worked on asymmetrical protein, and I think it was the first one to be crystallized in ribosomes, which is massive, about 500 amino acids, and they did it at 2 angstroms. This was a major advance in, it was because it was asymmetrical. This is a major advance in protein structure determination, 2 angstroms. The reason some of these are higher resolutions is because I don't know enough about this, but I think when the molecule's smaller, it's easy to get higher resolution. Okay, so this is amazing resolution. That's why I think, that's why his is low resolution. Smaller molecules, I think, is easier. But I'm not sure. Alan First, coming to modern times, Copley Medal, no, Royal Medal. He didn't get a Nobel Prize so far. He's developed atomic resolution folding of proteins, so down to the atomic scale, watching the atoms actually fold up. Carol Robinson is going in different directions. She's developed a completely new type of spectroscopy for whole molecules. Normally, spectroscopy involves heating them up and blasting them, or ionizing molecules and blasting them into millions of bits and then firing them through a magnetic field. She basically sent the whole molecule through a magnetic field. So she's been able to develop structural studies in flexible proteins as well as ribosomes, chaperones, which assist folding, and membrane proteins, which are all huge. This is a completely new area of spectroscopy she invented. And I should point out, I've heard her talking in the beginning. Nobody understood what she was doing. Nobody cared about what she was doing. And they actually fought her on it and said, why don't you just do what we do instead of wasting your time? But she's totally revolutionized spectroscopy. Royal Medal, Dame. Jim Barber, this is going a slightly different direction. So he says, this isn't pioneering work on big proteins, but it's more just succeeding in working out photosynthesis works. It's such an important process in biology. So in photosystem 2, he worked out the structure to 3.5 Armstrongs with so Iwata. And it gave him the first glimpse of the manganese cluster, which catalyzes the water oxidation. Manganese 3 Mn3 calcium cubic structure. And then these guys, a Japanese group, Suga, a little bit later, took it to 1.95 Armstrong, which is very high resolution. And then very modern times, you get Michael Hartmut in 1988. Well, it's not actually so modern, is it? But he developed a very detailed structure of plant photosystems, and in both plants and bacteria. They got Nobel Prize for that. And then Bill Rutherford in 2014 worked in photosystem 2 similarly, and showed the similarity between photosystem 2 and simpler reaction centers. So these Jim Barber was he was the first significant resolution of the photosystem 2 membrane protein complex, which was a major breakthrough. This was landmark. It showed the first glimpse of the manganese cluster, which is our Mn3 calcium cubate, which this catalyzed the oxidation of water. So this was a major advancement, which has been our holy grail in photosynthesis research for decades. These guys improved the resolution to 1.95 Armstrongs, but this was Jim Barber's. He wasn't the first membrane protein, but he was a major landmark in understanding how photosynthesis works, which is a huge, very important. Well, photosynthesis occurs all in nature. These guys actually were the very first membrane protein to ever be crystallized in 1988. They did it in 1984-5, which is bacteriorhodopsin, and they did full detailed structure of plants and bacteria. So this was actually the major breakthrough in doing membrane protein crystallography, which I thought had been possible before then. And this guy really was a landmark in understanding how photosynthesis works and the structure of the mechanisms of the machinery involved. And then Bill Rutherford just has improved it, has done modern recent work and similarity between photosystem 2 and simpler reaction centers. That represents his election to the Royal Society 2014, so it's fairly recent. So I've written there, none of these, well, they're pioneers of our membrane proteins. So when I say not pioneers, cross that out, they're all pioneers of membrane protein breakthroughs, and major breakthroughs in photosynthesis understanding. So they are pioneers of large proteins, but membrane proteins. So yeah. So over the last, I suppose, 100 years or so, protein crystallography has gone from insulin, which is 2 chains, and it was done at 5 Armstrongs. Dorothy Hodgkin, well, actually that was 2.8 Armstrongs, to Max Proust at 5.5. So they built fairly crude models to much bigger proteins, which are far larger, tens or hundreds of times bigger, which are now getting down to also around about 2 Armstrongs or just over it. So the advancements in crystallography of the last century have been absolutely they've gone from being pretty crude to right down to atomic level, where you can see the details of the atoms, which is amazing.